Hello, my dear students. Today we'll be talking about another topic that is your oogenesis. Yesterday we talked about spermatogenesis, that's sperm formation in case of the males. Now we'll be talking about oogenesis in case of the females. Now, oogenesis is the process of formation of female gamete, that is ovum, and is very different from spermatogenesis itself. So you should know that it is different from that. It begins at the embryonic life. Millions of oogonia, they are formed in the fetal stage itself, fetal ovary itself. So no new oogonia are formed after birth. Just remember that. Whatever oogonia have been formed before the birth, those oogonia will stay. Now, oogonia enters meiosis 1 and gets arrested in prophase-I. If we talk about more specifically here, it gets arrested at diplotein stage. Diplotein stage. And then forming the primary oocyte. Each primary oocyte is surrounded by certain cells which are called as granulosa cells. This is where, if you look into the diagram, textbook diagram, so these are the cells which are called as granulosa cells. They form the primary follicle. From birth to puberty, many follicles degenerate, and what is remaining at puberty is 60,000 to 80,000 follicles per ovary. Now let us talk about follicular development. Now, primary follicle gets converted into the secondary follicle. So what happens in the primary follicle, more and more granulosa cells will be added and one membrane theca will be added. So this is the transition from primary follicle to secondary follicle. Then secondary follicle will get converted into the tertiary follicle. And this tertiary follicle has antrum, that is cavity is there. Now this cavity is formed. And then theca differentiates into theca interna and theca externa. There will be two membranes now. Theca interna, internal, and theca externa, that is external. After the tertiary follicle stage, the primary oocyte completes the meiosis. Meiosis 1 I am talking about. It produces one large haploid secondary oocyte and one small polar body. This is unequal division. It ensures the ovum retains maximum cytoplasm nutrients. The tertiary follicle becomes the Graafian follicle. You can look here. And the secondary oocyte develops a zona pellucida layer. It's a membrane. Finally, the Graafian follicle ruptures and releases the ovum. This process is called ovulation. So this is actually what is mentioned in your textbook. So you have to focus on this only. There are different other things that are there. So you do not need to, we do not need to talk about those things. So whatever is written in the textbook, please focus on that only during revisions. So this is all about oogenesis. Now we'll be talking about this sperm structure and the difference between oogenesis and spermatogenesis. Thank you for watching these videos.